Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and CN Bio webinar titled It's Life But Not As We Know It, How Predictive Human Organ Models Are Transforming and Reshaping the Efficiency of Drug Discovery. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're being joined by Dr. Audrey Dubourg and Dr. Gareth Guenigault, respectively product manager and lead scientist at CN Bio, who will be exploring the value that an organ-on-a-chip approach can bring to drug discovery and development workflows, and describe how the regulatory landscape is shifting to encourage their widespread adoption. And with that, I'd like to welcome the first speaker, Dr. Audrey Dubourg. Audrey, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you, Liam, for the kind introduction. So before we start with the webinar, we have a first poll question. So what was your reason for joining us today for this webinar? So we're talking today about how improving organ models to improve drug development efficiency. Why is that? It's because one of the major challenges to any drug discovery program is the actual high failure rate of compounds. About 90% of the time, new molecule and therapy developed in pharma company fail to reach the market and fail in clinical trials. So why is that? It's due to two main reasons. First, unsuspected safety issues that are identified in the late clinical stage, or a lack of efficacy that is shown in the patients used in the clinical stage. There is also a case sometimes of drugs gaining entry to the market, but being withdrawn later due to safety reasons and patients' death. And that was the case for fialuridine a few years back. So scientists currently have a wide range of preclinical tools in their toolbox to help them evaluate new therapeutics and molecules. It usually starts with in silico, so identifying a few candidates for their target disease or a target projects, and then once they have these candidates, to move to in vitro cell culture, either monolayer or 2D culture, and then more complex 3D spheroids, to then move on to in vivo animal models. But all of those current preclinical models have one issue, is how well they translate to the human. And that is mainly due to quite a few factors. Those current methods lack human relevance, especially for the animal models. As we can see here on this table, we do have a few animal models. So animal models do not have any human relevance because they are other animals. There's other genetics and immunologic effects as well. The in vitro cell culture models currently used most of the time use either immortalized cells or iPSCs, which are not that great. The complexity of the model in vitro lacks the three-dimensional effect of the organs, whereas animal models do have a full 3D organ, which is great. Animal models also have a blood flow inter and intra organs, which really recreate the complexity of an organism, whereas the current in vitro models do lack these effects. The immune system can also be implemented in the animal models because they have their own immune system, whereas the in vitro cell culture models are lacking that. The longevity is also problematic, especially for the standard cell culture, which usually goes not longer than 7 days because the model either lose their functionality, the cells dies, or they de-differentiate with their phenotypes. Whereas animal models can go over 4 weeks, so we can have really long-term studies to study the long-term efficacy or effects on the organism. Only animal models offer acute and chronic dosing. However, the throughput is very low. It's one animal, one condition, where the current cell culture model do offer really high throughput, so we can get quite a lot of answers before moving to animal models. The ease of adoption can also be problematic with those ones. Animal models require very skilled and trained users or technicians and researchers to use them, whereas cell culture is a bit more straightforward. We don't need any particular skills. bit of adaptation for 3D, but overall better than animals. The cost is very high for animal models between the fact that we need to breed them, to feed them, to host them, and then run the assays, whereas cells are very much cheaper. The time to results is usually fast for cell culture, which is great, but we get a very low to medium data content generation, whereas animal models are very slow, but we will get a very high content data generated. So what is done currently to help compensate that issue in terms of translation to the human for those preclinical tools is the use of new alternative methods. We hope that those new methods, also called NAMs, will help bridge the gap between current preclinical in vitro and in vivo models. And with the development of NAMs, and the failure of drugs because of poor in vivo data and poor in vitro data, we are seeing also a growing pressure for change to the regulatory landscape. So, for example, back in 2014, here in the UK, plans were announced to reduce the use of animal tests in scientific research, aiming to replace those tests with scientifically valid alternatives wherever possible. More recently as well, in 2021, the European Parliament voted in favor of plans to phase out animal testing in research. And also in 2021, last June, the US Food and Drug Administration, the FDA, set out proposals for the NAMS program to focus on replacing, reducing, and refining the use of laboratory animals through the adoption of cutting-edge alternative methods. So the aim is to produce really findings that are more relevant to humans, streamline product development, and reduce costs. That shift, which has been years in the making, would be undertaken across all of the FDA centers, including the ones that are overseeing the approval of new pharmaceuticals, medical devices, veterinary medicines, cosmetics, and more. To add on to that, this year, the FDA Modernization Act was passed by the US Senate and is currently awaiting approval by the US House of Representatives. And that will enable researchers to circumvent animal testing for human relevant and predictive methods to also develop the next generation of therapeutics. And when hopefully this bill is passed, that will end the very outdated FDA mandate that requires experimental drugs to be tested on animals before they could be used on human in clinical trials. So really, researchers will be given options to use scientifically rigorous, proven non-animal test methods when they are suitable. However, in the interim, while the House of Representatives is discussing that, drug efficacy testing is currently not regulated, so alternatives can be used in this space. Interestingly, we're also seeing more and more new human-specific modalities within the pipelines of drug discovery companies, such as cell and gene therapies. And in this space, there are few, if any, preclinical in vivo models available to predict human efficacy, toxicity, and drug disposition. So here, more human-relevant models are required to address these unmet needs, and this is what NAMs are set for. And when building those NAMs, many factors have to be considered when building them, and we need them to be reliable and robust, so the technology, but also the model, to have a clear cost that is easy to implement. Are there any clinically relevant translational endpoints? Do they translate to the human? The throughput as well is very key, how representative of the human biology they are as well. Once those questions have been answered, we get into more of a, can the model used mimic the in vivo pharmacokinetics of the compounds? Can we do acute versus chronic dosing? Is population variability something that can be studied as well? And can the immune system be involved to study immunological effects? And one important thing as well that we keep in mind when we develop those assays nowadays is how we can study the effects of new modalities such as gene therapy agents, as the current methods are not fit for purpose there. One of those NAMs that has been developed in the last 10 to 15 years are organ on a chip. There has been a growing interest in organ on chip ever since the early noughties, with first the development of macrofluidics devices to help with the cell culture in academia mainly, And then in 2009, the first ever publication on lung on a chip by Harvard University that really bridged the step forwards for organ on a chip and all of the new technologies being developed nowadays. And we see an increased exponential interest in those systems ever since the early noughties. So what are organ on a chip? They're also called microphysiological systems, or MPS. Those systems come in many different shapes and sizes, but they all have some common features. First and foremost, the use of human cells and tissues. They are typically grown in 3D and with some geometrical confinement or patterning to really help the recreation of the macroarchitecture of the organ targeted. They also need a fluidic flow to mimic the blood flow, provide oxygen and nutrients, and also provide some biomechanical stimuli to recreate the shear stress those tissues and organs are under in an organism. Environmental control is also very key. Can we put any mechanical actuation? Electrical stimulation is important. For example, for a heart model, we do need contractility, so the muscles to contract and really recreate that function of the organ, but also the control of gas exchange and any growth factors needed. The last one is the drug dosing. Can we control the exposure to new therapeutics? Can we improve the way we monitor and measure this response with built-in electrodes or sensors or real-time monitoring in there? Organ on a chip can be used in a wide range of different phases in the drug development process. They are mainly used for now in the discovery phase, where in target and lead discovery for disease modeling, they can help with understanding cue signal response, any kind of fluidic and mechanical stimulation of disease states, but also help with the basic research in terms of targeting and target identifications. When we move a bit further down the ladder of the drug development pipeline, we have the DMPK safety, where by using multi-organ, we can study the inter-organ crosstalk, oral bioavailability, any kind of inflammatory responses as well, toxic response. And when we move really close to the end of preclinical to clinical, confirm the target, the drug efficacy on the target organ, but also the link to any toxicity for that organ or any off-target as well. In the development phase, they will be used more to generate more data in terms of investigational toxicology when we want to have more data to supply with the IND submissions. But if a drug were to fail as well in the clinical trial, organ on chips could help with the repurposing of those drugs too. So there is an extended kind of application areas for those organ on chips in the drug development pipeline. So if we go back to our previous table in terms of what are the limitations and what can organ on chip do compared to the other preclinical tools, what we see is that organ on a chip are very human relevant. Most of them use primary cells, and therefore they give that edge that we need to really match the human physiology. They are more complex than the standard in vitro cell culture because we do have some flow, we have structure to create the 3D models. They're not as good as a full organ like in the animal models, but they still compensate a lot of the issues we see with the current cell culture models. They do have flow perfusion, so recreating like the systemic circulation. Some of them can be interconnected organ as well to really help study inter-organ crosstalk there to kind of recreate part of an organism. Some platforms allow for the immune system to be added, so either circulating cells or none, which helps with having an actual human immunological response. The longevity is on par with animal models, with about 4 weeks for a study. Acute and chronic dosing can be done as well. Depending on the platform using, the throughput can be good. Some are lower throughput because they're more high-end. So it really depends on what you want to do. The ease of adoption is also somewhat okay, depending on the platform you want to use. This can be more complex with very like proper chip versus plates. The cost will greatly depend on the platform, but it's still an intermediary cost between animal models and cell culture models. But what is really important is the time to result is very fast because you can get a lot of outputs out of the cell culture during the experiment and at the end with the tissues, and the data generated is of high content as well. So quickly before we move on to the next step, we have a poll questions. Do you currently use 3D models? So organ-on-a-chips can be either used by researchers by acquiring platform and putting them in the lab, or to be used as a contract research. That's what we do at CN Bio. We offer you the best of our technologies the way you want them, and for applications area in disease modeling, safety toxicology, and ADME mainly. And all of that is powered by our system, the PhysioMimix. The PhysioMimix is that big system you see here at the bottom with that big white box being the controller that sits outside of the incubator. It controls everything that happens in the incubator by pneumatics and electrical cable here, connected to that docking station at the bottom here, on which an MPS driver and an open well plate will be sitting, and where the biology will happen, and the microfluidic as well. The system works currently with 3 different types of plates. We have a dual organ for multi-organ studies, so lung and liver, or gut and liver. We have a barrier plate as well that enables the study of barrier models such as a lung or gut on their own. And we have a bespoke liver plate to study healthy or diseased liver, and we're going to be focusing on that one for the rest of this webinar, as the liver has been well characterized in our platform. So to run a liver experiment, we simply select the cell type we want. We usually use primary hepatocytes. And then if we want to use non-parenchymal cells, we're going to add them depending on the applications. You can use Kupffer cells or stellate cells. We're going to add the liver media from day one to grow the tissues. If there is a disease states that you need to induce, we start from day one onwards. And then the real experiment will start from day 4 when the tissue is fully grown and metabolically active. We're going to start to do a drug dosing for however long we want it to be done. We have gone over 28 days. Some customers have gone over 32 days as well. And the data generated are from the media, the tissue structures, and if you have any probes to be put in it from non-invasive measurements. The data are generated through any kind of standard lab experiments, so any omics, microscopy can be done on the tissues, histology as well, and biomarkers and metabolites for the soluble markers there. So once the experiment is ready, how does the fluidics make the liver tissues? It's a simple fashion. In an open well plate, we have 12 independent wells in which a bespoke liver scaffold is sitting at the bottom of the well. You can see here. Cells are seated on that scaffold, and the flow will push the cells inside of the microchannels inside of the scaffold. The flow happens just simply because you have a membrane there sealing your plate and air being pushed in and out, forcing the liquid on top to actually circulate in your plate. And the flow rate can be adjusted to the need of your experiment. The tissue generated via morphogenesis from the single cells to a complex microtissue here. You can see here the tissue is polarized. We have tight junctions between the hepatocytes, and we have formation of bulk caneliculi. If we focus more on the bulk canaliculus here, we can see that we have a formation of macrovilli, and the lumen of the bulk canaliculus is also present with the tight junctions. So this liver macrotissue really recreates the liver macro architecture. That tissue is highly functional for a long period of time. We see secretion of albumin and even urea over time that is quite steady. And we have low level of LDH, which shows that the tissue is healthy. The tissues are also metabolically active. We see a lot of CYP enzymes being secreted over time, and using different donors, we can really study the donor-donor variability in the model. Another things we do to develop our assays is qualify donors, because it's really great to have a great platform that is easy to use and a model that is well defined. But if the donors are not great, we're always going to struggle. So to do that, we select donors according to their age, pathoviability, the lot size, the genetics, but also do they qualify for 3D cell culture. We're going to sit them in the plates, run an experiment for 2 weeks, and then we're going to measure the microtissue formation. So here we have an example of 5 different donors, with B and C being the best donors. We have a nice tissue formation, E being the worst. We barely have any tissues, more of a lot of cell death. We're going to also measure the CYP activity for all those donors to check how metabolically active they are. The albumin production to see if they're functional, the viable cells per vial, the more cells the better, but also the well-to-well consistency is very key to be able to reproduce those tissues in more than one well. Before moving on to the next step, we have another poll question for you. How familiar are you with organ-on-a-chip technology? And now I'm going to hand over to my colleague Gareth Guenigault, who's going to talk to you about a couple of applications and case studies in our liver plates. Thank you.
All right, thank you very much, Audrey, for the introduction. So yeah, I'm going to be talking about a few different case studies and essentially ways that we use our liver plates and sort of the models we've created on that system. So the first one I'm going to talk about is modeling NASH. So very quick summary on NASH, non-alcoholic steatohepatitis. This is essentially the advanced form of non-alcoholic fatty liver disease, and is characterized by the fat buildup in the liver, which starts to cause inflammation and fibrosis, and that can be very damaging to the liver. And it's essentially an unmet medical need. There's a lot of compounds that have been developed for this, but none have really been successful so far. There's been some fairly big failures in the clinic, and clearly the efficacy of these compounds is not being captured properly in the current preclinical models. So our NASH model has a co-culture of 3 different cell types. So we've got our primary human hepatocytes, which is the main cell type of the liver. We have Kupffer cells, which are the sort of resident immune cells, and a lot of the inflammatory part of NASH will be through those cells. And then we have the human hepatic stellate cells. So they are responsible for a lot of the fibrosis that is seen in NASH as well. So we culture those 3 cell types together, so into each of these wells. And then the day after we seed those cells, we change the media into what is essentially almost like a high-fat diet. It's a media containing high levels of fats and sugars, and then that starts to induce that NASH phenotype within those cells. We then leave the cells for a few days to be able to establish themselves and to actually kind of form that NASH phenotype. And then we can start testing any therapeutics of interest. And as I said, there's a range of different models here in terms of the severity of the disease that we can create. And that's been well published on now in a range of different publications. But the one I'm going to talk about now is this lowest one. Yeah, that's a collaboration with AstraZeneca. It's modeling human liver fibrosis in the context of non-alcoholic steatohepatitis using a microphysiological system. So there were a few aims for this project. The first of which, well, set the scene a little bit first. We'd already at this point actually developed our fat media. We'd already decided on the triple culture of cells to go in the system and the general sort of timings and length of experiment. But we wanted to see if we could alter it in other ways that would be interesting. So what we were doing here was treating with endogenous ligands such as TGF-beta, LPS, fructose, or cholesterol, or a combination of those to determine what kind of effect they would actually have on the NASH phenotype. We also explored the transcriptomic profile of the model with or without these different additives, again to see how relevant it was essentially to human NASH. We also want to look at new ways of being able to explore our assay and the actual phenotype that we were developing. And so we were working on confocal imaging of fibrosis, looking at key markers of fibrosis within the cells. And then finally, exploring the actual pharmacological response within the system. How did the model actually respond to treatments with various compounds? So on the bottom here, you can see the sort of standard NASH experiments, so the general layout and timings we used for all of the experiments we're going to talk about here on the NASH side of things. So as I said, we seed the cells on day 0 on that triple culture, change to fat media the following day, and then leave the cells until day 4 to develop that NASH phenotype. And it's then that we start the treatment, whether that be the TGF-beta, the LPS, or the compounds that are fibroid beta-cholic acid. And then the experiment run up to 14 days, so essentially 10 days of treatment, and see what effect that had. So the first part on altering the NASH phenotype, I've got 2 sets of data here. The one on the right is essentially exploration of some of the transcriptomic changes. So the differentially expressed genes between essentially a healthy condition or the NASH condition with all those various ligands. So you can see here treatments with fat, LPS, cholesterol, fructose, and TGF-beta, and the different pathways involved, and essentially how they change in terms of those specific pathways and the levels they're changing. On the left, you can see Luminex assessment of the cytokine profile, and again, with those various different treatment options. So there were some very clear and obvious changes between some of these different treatment options. LPS is an interesting example. As you'd expect with something like LPS, there's a very strong inflammatory profile. As you can see by far, which is row 6 here, or column 6, it's got the highest cytokine profile of any of the options we did. However, there's some interesting effects here. So this is column 3 for this side of things, and you can see there's no real change to the extracellular matrix organization, and a lot of changes in areas such as interferon signaling and complement. So even with the fat and the LPS, you get a really big cytokine response, inflammation response, but it really doesn't seem to have as much of an effect on the fibrosis, even as, you know, in comparison to just having the fat media on its own, which is quite interesting to see. The TGF-beta, on the other hand, as you'd probably expect, had a massive change, as you can see down here, in a lot of the kind of collagen-related pathways of fibrosis. So regardless of whether it was TGF-beta plus any other of the treatments, you were always getting this big change here. So this is any TGF-beta plus other bits, essentially. And again, a big change in the extracellular matrix side of things, so really driving that fibrosis response. And you're still seeing some changes occurring as well in the inflammatory profile. Now, other treatment options like cholesterol, interesting in their own rights, but say, example, cholesterol here in this first column really didn't seem to have much of an inflammatory response, which is interesting, did show some fibrosis. And then fructose as well had some effect, particularly some effect in the inflammation, but worked better again with TGF-beta. So a lot of what we took from this analysis was, you know, it's not necessary that any of those treatment options are right or wrong. It's more that it's very helpful to understand what question you're trying to ask of the model. Say, for example, you actually have a compound that really wants to affect inflammation, then you might want to run the system with LPS in it and see that actual inflammatory response to give you a bigger window to be able to knock down some of that cytokine profile. A lot of people are more interested in the fibrotic phenotype of NASH. That's often one of the key endpoints people are trying to reduce. So for that reason, and some other bits I'll come to, a lot of the work we've done going on from here has been either the cell triple culture with the fat media, and then with or without TGF-beta to kind of get that real fibrotic response. So the other part of this assessment, and particularly again looking at that transcriptomic profile, was to kind of explore what disease pathways were coming up. So we basically compared the differentially expressed genes from our model to the human gene atlas. And the very nice thing to be able to see here was that the main pathway it came up with, the top pathway, was NASH. So that gave us some good early indications that our model was capturing the actual human disease. The other side of this is we compared the differentially expressed genes in our system to those seen between sort of healthy and NASH people. So essentially data from patients, and we see a very, very nice overlap with our data to that data. So you get about 50% overlap compared to murine models, which tend to sit much, much lower than that. I would also say that that kind of analysis has been replicated by other groups. So Michele Vacca did a really nice piece of work doing a similar kind of differential gene expression analysis and showed similar data. So I recommend you look at some of the work he did as well. So having kind of gone forward and created this model and, you know, decided that, you know, it's either the NASH, which is the fat treatment on its own, or NASH plus TGF-beta model were kind of probably the most relevant and most useful, we wanted to find new ways to be able to look at our system and actually measure the kind of phenotype changes. So in particular, as I said, fibrosis being one of the really key hallmarks of the disease, we wanted to look at confocal microscopy and see how the fibrosis changed visually, but not just visually, we also wanted to go and quantify that. So here you can see NASH or NASH plus TGF-beta, and we're looking at collagen, smooth muscle actin, and then just Hirsch as a nuclei stain, and then a merge of the three. And you can really see a very clear, almost change in the way that the cells behave with the treatment of TGF-beta. You get almost sort of striated cell structures occurring as those stellate cells really become overactive and start producing a lot more of that collagen and alpha smooth muscle actin. Now, obviously, really nice to be able to see that, but we can also look at it in terms of data, get some numbers on here. So as well as looking at fibrosis through confocal microscopy, we can measure secreted markers in the media. So this is TIMP1 and procollagen, both, as I said, detected from the media. And you can see a significant change in these two. And then what we were really hoping is that we could essentially find data that correlated with that from the imaging, and we could. So here is the quantification of the smooth muscle actin and type 1 collagen as taken from the imaging that we'd assessed doing the two different models. Okay, and then the final stage of this really is, very nice to have built a model that seems to replicate the key stages of the disease and really, capture that human phenotype. But, can we actually reduce the NASH biomarkers using compounds? So here we've got two therapeutics, elafibranor and obeticholic acid. At the time, they were the leading clinical candidates. Now, both of them got to phase 3 clinical trials, and we tested them in our system. And essentially, at 3 different concentrations, you can see this dose-dependent response, where you get the reduction in collagen, as you can see in obeticholic acid here, and elafibranor, and smooth muscle actin, and then the quantification of that as well. You can see this gradual decrease as the concentrations increase. So to summarize that piece of work, really that treatment with the exogenous ligands can dramatically affect the NASH phenotype. So we've got a system here that's quite flexible, and if there's a specific question being asked, it allows us to be able to adapt the model slightly to suit the needs of the person doing the research. As I said, inflammatory response, you may want LPS there. If you're really focused on fibrosis, then TGF-beta might be more useful. We selected the NASH model with or without TGF-beta as our kind of most human relevant models. I would say that essentially what we see is the model without the TGF-beta is somewhere around, if you look in the fibrosis scores that are often used, it's about an F1, F2. If you're treating with TGF-beta, that ramps it up more to an F2, F3 kind of state. So yeah, that can be quite useful to sort of understand. We can do biomarker analysis through both secreted and cellular endpoints. So secreted side of things, we very, very frequently do ELISAs or other measures from the media. Every time we do a media change, you can essentially collect about 1 milliliter worth of media, and we do that every 2 to 3 days. So that gives you a wealth of material to be able to assess and explore the response, anything that's secreted. So Luminex, lipidomics, proteomic analysis for what's going on in that media is very helpful. And then obviously being able to assess the cellular endpoints and develop systems like confocal microscopy is a very useful way to kind of be able to assess things both visually and to be able to quantify that. And, everybody likes to be able to see the changes they're exploring. So that's always a very powerful tool. So the NASH model is an excellent model to be able to test therapeutic responses, and it allows us to really explore a range of different hallmarks of NASH, such as inflammation, fibrosis, and steatosis. And crucially, you can do that using human cells to replicate human tissues. So final poll question, I believe. What is the biggest challenge you face with your 3D cultures and experiments? So the next case study I want to talk about is using our liver chip for basically looking at safety and efficacy within the liver. So in this model, we tend to do a co-culture of primary human hepatocytes and Kupffer cells, so we don't have the stellate cells present. We culture those in, to be honest, fairly standard media, so that you've got these kind of nice healthy conditions. And then really what you're doing is you're adding in your therapeutic, and that will be what causes the actual drug-induced liver injury. And so this model really allows you to explore, this kind of toxicity profile of what you're looking to test. The other side of this is it also allows you to look at the metabolic profile of the compound that's being tested within the system as well. So I'll talk about both of those aspects. Again, this is a well-published model, and I'm going to focus on this particular paper, which is a collaboration with the FDA, which was characterizing the reproducibility in using a liver microphysiological system for assaying drug toxicity, metabolism, and accumulation. So again, there were a range of different aims that we were looking at in this particular piece of work. So the first was really to do a comparison of different liver tissues and different models. So our liver MPS model versus spheroids or 2D sandwich cultures, all of them using primary human hepatocytes and Kupffer cells. We were also interested in seeing, firstly, could the model be used to capture key aspects of drug-induced liver injury? But also crucially, could that be captured, and did you get similar results at multiple sites? So, it's kind of that ease of adoption kind of question. There was also a comparison of hepatocyte DILI response in multiple models. So again, going back to that, how does DILI effects occur in our MPS model versus spheroids and the sandwich cultures? And then finally, doing a bit more of an exploration of the efficacy of drugs and their metabolism profile and accumulation within the cells. So what we can see here is essentially a month-long experiment exploring the activity and stability of primary human hepatocytes and the Kupffer cell co-culture within each of these different systems. So we've got blue, we've got our liver MPS, pink is the spheroids, and yellow is a sort of standard 2D sandwich culture. So first you can see the metabolic activity. So this will be through CYP3A4 activity. And you can see the liver MPS as a very stable long-term expression and of an activity of that particular CYP enzyme. It does drop gradually over time, but it's still very much active towards the end of the experiment. In comparison, the spheroids, while they do maintain some level of activity, it's much, much lower than in the MPS system, and does drop off that little bit earlier because it's got the lower starting point. As well as that, the sandwich culture, while it starts high, drops off very, very rapidly. So really, by day 10, your culture is no longer really active anymore. And then albumin production, which is a really good marker of your liver cells being functional and showing that they haven't de-differentiated. We can see here again, nice high response albumin production over time using the MPS system. The spheroid one, while it is still detectable, it's very, very low in comparison. And the sandwich culture, again, by day 10, I mean, to be honest, earlier than that, it's not really producing any albumin. So that it looks like the cells are more likely to have de-differentiated and your cells are no longer active. So yeah, so we believe our model here is far more active and you've got much better production. You're keeping basically a nice stable culture of hepatocytes for a longer period of time. Then looking at the DILI response, so the data I'm showing you here, there's an 8-day cell culture to establish the cells followed by a 3-day treatment. What we're looking at here is trovafloxacin and levofloxacin with or without LPS. Now, trovafloxacin is expected to show some idiosyncratic DILI, whereas levofloxacin is expected to essentially be well tolerated by the liver, as seen in people. And there were multiple sites here to look at as well. So we've got LDH data in the top row, which is essentially showing if there's any toxicity occurring. CYP3A4, again, looking at that metabolic activity in the lower row. Site A was any work performed at CM Bio, and Site B was work performed at the FDA. They also decided to test 2 different Kupffer donors, whereas these 2 bits of experiment use the same Kupffer donors. So the key things we can really see here is that generally we are seeing the same response, which is that it's the trovafloxacin with at 100 micromolar with LPS that gave the highest kind of toxic response and release of LDH. So you can see that here, and actually coming through to the second Kupffer donor as well. There is some slight differences occurring, particularly between the 2 Kupffer donors, in terms of a slightly higher LDH release in general, particularly in any treatments with LPS. And then CYP activity follows a very similar profile, essentially showing that actually trovafloxacin with or without LPS completely really abolishes the CYP activity, and that was captured at both sites. But again, the 2 Kupffer donors behaving slightly differently in that any LPS treatment really seemed to affect this donor and reduce the CYP activity, which was interesting to note. And it does highlight the importance of making sure that you test and select the right donors for an experiment to get the answers you need. So also following on the DILI response, we also want to explore how different models were affected using specific compounds. So this is a 6-day cell culture followed by 2-day treatment, and we were looking at our own MPS system, again against spheroids and 2D sandwich cultures, and we were testing triglycerin, tamoxifen, and digoxin at 7 concentrations each. Now, what you'll be able to see from all this data is that the toxicity is essentially observed in all the models, but the sensitivity can be quite different. So we've got LDH release here in the pink, which obviously starts to increase at the higher concentrations of triglycerin. The CYP3A4 activity in the yellow decreasing, and the albumin production, to be honest, following a very similar profile to the CYP3A4. And while in the liver MPS, it's well over 100 micromolar before you really start to see the severe toxicity. In the spheroids, it's a bit below 200 micromolar, and in the sandwich culture, it's quite below 100 micromolar that you start to see that toxic effect. And that follows through for the 2 other drugs as well. So it's really important to kind of understand how your compounds or your therapeutics are going to behave in each of these different models, and actually moving between any of these does require potentially a relook at the actual effective concentrations or toxic concentrations that you're exploring. And the final piece of work to highlight from this paper is looking into kind of metabolite formation, the metabolism of the system. So again, it's the same co-culture of cells, an 8-day cell culture followed by 2 days of treatment. And what we can see here is treatment with triglycerin at 50 or 100 micromolar. And really what we want to observe here, and what we did observe, is, as you'd expect, at 0 hours, you're getting a lot of triglycerin within the system. You've only just added your drug. By 48 hours, as measured from the media, you are essentially, there's barely any of the triglycerin left within the system. So there's no media changes occurred here. That is drugs all being metabolized and broken down by the cells. If you look at the 2 phase 2 metabolites we were looking at here, as you'd expect, none of it at 0 hours, but a very nice concentration of both of those by 48 hours, again suggesting that our system has metabolized basically the parent compound into these 2 metabolites. Very, very useful to understand the profile of different compounds and how they'll affect, how they'll work in different systems. It's often those metabolites that are toxic to the system. To be able to understand the kind of ADME profile can be really, really useful. And then the other side of things is looking at intra-tissue accumulation. So this was looking at chloroquine, which is known to accumulate within the liver. And essentially what we're doing here is measuring chloroquine from the media or from the actual tissue lysates. And what we found was a 5-fold higher concentration after 48 hours of treatment within the tissue lysate than in the supernatant, which is showing that the chloroquine is actually being taken up and accumulating within the liver, again replicating what that compound does in people. So to summarize that particular piece of work, we found that the PhysioMimix liver MPS is a very long-term system. So it allows you to culture cells that are very stable. They have high expression of hepatic biomarkers. They've got good metabolic activity, and that really allows you to explore compounds that might not have an effect over a 7-day period. They need longer treatment periods so that you can actually see their efficacy. The NASH model is a good example of that, actually harking back to the earlier data where, say, for the elafibranor, you don't really see anything until day 14. So you need that 10-day treatment period before you're really starting to see the effect properly, particularly on fibrosis. There are key DILI responses that can be captured within the system, and that can be replicated across multiple sites. So one of the things Audrey was talking about, we have this nice open well plates, means it's quite adaptable. They're relatively simple to use, particularly if you're familiar with cell culture in general, and that obviously helps the kind of adoption of the system and ease of use. It was interesting to note that you get different sensitivity between different model types. I'm often asked whether, people come to us wanting to test a drug and say, well, I know where it's toxic at in a 2D culture, but that might not be, as you can see from this data, where you'll see the toxicity in our model. And so it's still very useful whenever you change any model type to still test a good range of compound concentrations to ensure you get the best data. And then finally, that our PhysioMimix liver model has a high metabolic activity, And that really allows you to predict the compound clearance and metabolite formation over time, which can be exceptionally useful. So for a summary of everything, what organ-on-chip really enables you to do is closely model human physiology, and that's really the crucial part of what we're trying to do here. And that allows you to more accurately predict human responses to drugs rather than current preclinical models. You also have better in vitro to in vivo translation. You're able to use clinically relevant biomarkers, actually something I haven't presented here, but there are other markers such as ALT and AST, which are often used in clinics, that can be measured and detected within our system, and we can see changes of those with toxic compounds. And essentially what this all means is that, we're getting decreased risk of unexpected results in the clinic. There's a lot of compounds, as Audrey was talking about, that they look like they're efficacious, they look like they're not toxic when you're using animal models, and then you put them in clinical trials and they're very expensive failures because they haven't captured the physiology of what's going on. And so we're not saying at this point, get rid of animal models. What we're really talking about is, use your animal models. You've got that in vivo data, but you should really at this point be looking at having advanced 3D models, having organ-on-a-chip style technology to be able to provide that human component to the data you're looking at. So you've got the combination of your animal data and your human data would actually provide a very, very, very powerful tool to help get your compounds through clinical trials, which helps fast track that kind of decision-making process and ideally get your compound and your therapeutics to market quicker. So that's everything from me. Thank you very much. And I think we're going to go over to questions now.
All right, fantastic. Well, thanks so much, Gareth and Audrey, for the great presentations. I will now be moving into a Q&A session. So if you do have a question that you'd like to ask, definitely submit it in the Q&A tab on the right-hand side of your screen. All right, first question here. So in the NASH model, you only show elafibranor and OCA data, but have you also tested other anti-NASH compounds or new drug modalities on your platform?
Yeah, no, tested both of those two. To be fair, they're still two of the most advanced compounds that have been tested in patients within NASH. So there's a wealth of information about them, which is the main reason we selected those. The short answer is we have tested other things. A lot of that has been in projects for customers, so we can't really share the data as much so easily. But we've tested things like ALK5 inhibitor, which seems to have worked quite nicely. And basically, we're always looking at new compounds to be testing as well. So we've got ongoing projects kind of trying to extend that portfolio of different compounds we've tested within the system.
Yeah, fantastic. Great answer. So next one here. So this person says they currently use 2D culture. How easy is it to transition from 2D culture into a 3D, the 3D PhysioMimix assays?
So it is fairly straightforward when using our system because it still uses an open well plate. So most scientists are comfortable with it. It's still the same setup. There is a bit of transitioning in terms of how you handle the plate because there is some macrofluidic in the system, but it is not too cumbersome. So you don't need to be highly skilled. Like for some of the other organ on chip out there, that is a very tiny chip where you need to really be careful how you handle your cells and your platform. So it's fairly straightforward to set up and get on with a proper 3D knowledge with it.
Fantastic. All right, next question here. Does the CN biosystem support high throughput screening of drugs and small molecules?
I'll take that one as well. As Gareth showed, and we haven't shown the data, but we have we have done work with small molecules like ASOS, and Gareth mentioned others for the NAS project. So currently our system setup is not high throughput, so it's more medium throughput because you have 12 wells per plate, and a system can run up to 6 plates, so you can go up to 72 really conditions or replicates depending on what you want to do on the system, which means you can still do some kind of medium throughput in it, but it's not high throughput like you would see in a 96 well plate or even more. So the thing is you have to compromise with the macrofluidic in those plates in terms you have to either sacrifice on the level of throughput or in terms of scaling for your tissue and the volume of material you want. But it doesn't mean we're not trying to improve that for now. We are still working in the background on improving those kind of hurdle to give organ on chip early access for like researchers as well, so they can use it earlier in the preclinical and drug discovery pipeline.
I would also just quickly add to that to say that it is, while it's not super high throughput compared to some of the other organ on chip models, it's got a high throughput as well, and it is very, very standard for us. We've got enough wells present that we can work on that we do, triplicate testing of any particular condition or even quadruplicate. So it's enough to be able to have those replicates to make sure you've got the solid data set.
Fantastic. All right. There's a good question here. Does the system work with iPSC derived hepatocytes?
Short answer on that one is yes. I would say that we've not validated it as well as we have the primary cell model, but we can certainly grow the cells in there, demonstrate, tissue forming and, production of what you'd expect from hepatocytes in the model. But iPSCs also have their own flaws, as all the different cell models do. So we've chosen mainly to rely on primary human cells, and that's the main model we've worked with. But you can do it.
Fantastic. All right, another one here. Can this system replicate liver endothelial fenestrae?
I'll take that one if you want, Gareth. It's a tricky question because Gareth showed you a co-culture with Kupfer and hepatocytes. So the answer for that one is most likely no, because we don't have endothelial cells. We currently have only developed up to 3 cell types in it, with the stellate cells as well for the fibrosis in NASH. So short answer is for now, we don't know, because if we were to add endothelial cells, we would need to assess that if it's possible or not. But currently in the models we offer, it's a no.
Right, makes sense. Can your liver model be used to study low clearance compounds?
Yeah, that's a definite yes. So that one of the advantages of having the kind of longevity of the actual cells lasting for a month long essentially allows you to be able to do longer basic periods of testing things than you normally would. So obviously the key part of doing the low clearance side of things is how long the cells survive without any media changes, fresh media or anything being added onto them. And we've certainly done experiments with essentially about 4 days worth of length without changing the media at all and still having good functionality and being able to detect some of these sort of low clearance compounds as they are being sort of slowly metabolized and cleared from the system. So yeah, that is put within the system.
Excellent. Yeah, great to hear. Can any organ be recreated in vitro using organ on a chip technology or MPS? And what other organs have you developed using the system?
So in theory, yes, it is possible. But however, the type of platform, the type of support, and the type of fluidic flow you'd need for each organ has to be really tailored to the organ you're targeting. So it is very challenging. Some organs still don't have we don't have access to any kind of good primary cells for it. It's such a niched environment that suppliers don't give those as well. So there's a lot of work to be done. In terms of all the organs we have developed in the system, we have lung models using primary cells, and we have a bronchial and alveolar model for that, quite complex and quite nice. We have some application, also webinars, people are interested in it. We've developed a gut model before using what was accessible. So we started with KCOS and HT29. We are working on assessing how to develop a primary gut model to have something that is more relevant to the human, because immortalized cells are always limited in what they can give us. And we also can link those organs to the liver with a multi-organ. We have a lung liver. We have currently the current gut model connected with the liver to really increase the studies with the organ crosstalk and to get through the oral bioavailability as well of a compound and see how the metabolism goes through the absorption as well and everything like that. So yeah, I think the main answer is you could develop any organs, but there's a lot of work to be done. And so that's here we go.
Yeah, definitely. It's a really exciting area of technology right now. I think a lot of work is going into it, which is really fun. So next questions just come in that maybe lets you brag a little bit, but what are the advantages of the PhysioMimix system compared to your competitors?
So it really depends on what type of platform you're looking at. If we really focus on proper organ on a chip, so the really small chip that you have from different competitors, it's first the initial setup because it's very easy to set up. I'm a microbiologist by trade and I can do it in 10 minutes. So it's easy to set up your system and transition to it. The throughput compared to those is also quite important because we can run, as we said, 12 wells in one plate. So if you do a triplicate, you can have 4 conditions in one plate. So that reduces the cost of your experiments as you can run more replicate in one plate, as those chips are quite expensive. It's also in terms of volumes of and tissue scales you get in the system. As we get up to 1 milliliter of media every time you sample, you can get a lot of biomarkers analysis or anything done on endpoint like that. And the tissue is big enough that we can split the scaffolds in half and run microscopy and any kind of omics on the tissue as well at the same time. So you get a lot of answers with one well that you would need several chips for. Another advantage is because the system is a closed system with the liquid recirculating in it. We can also add immune cells, circulating immune cells like PBMCs or LTs or everything like that in the system to really study any kind of immune-mediated responses in the system. I think that's the biggest one on top of my head. There are probably more, and I could find them if we had the time, but I think we can answer more questions.
Perfect. Yeah, I think we do have time for a few more. We actually did have a question, but it sounds like you just addressed it. But maybe you could elaborate a little bit. The question was, can you customize the PhysioMimix models, for example, by adding in extra cells like circulating immune cells, exactly as you said?
Yes, maybe I'll let Gareth answer a bit.
Yeah, of course. No, so one of the nice things with this is, everything's kind of the structure of the plates, for example, you've got a lid and everything. It's kind of familiar to how you do like a standard 24 well plate in terms of scale. And that means it's quite flexible. So you can add the specific cell types you want into. So, for our liver model, we do hepatocytes on their own, hepatocytes plus Kupffer cells, hepatocytes plus Kupffers and stellate cells. There's no reason we couldn't add the endothelial cells into there if we wanted to go down that route. I suspect we probably will be doing that as a task in sort of the next set of experiments. And then immune cells are certainly able to be added into the system as well. It's an advantage of having that circulating media. You can add the immune cells in. You can see how they flow and go past. You can look at how they can, attach the other cells present within the system, the effect they have on the kind of immune response in your model as well. So yeah, no, that's it's certainly possible.
Great answer. All right, short question here. Are your plates made of glass and are they disposable or reusable?
I'll take that one. So the plates are made of plastic, like cell-culture plastic. It's called COC, which stands for cyclic olefin copolymer. It is currently the material available with the lowest non-specific binding properties you can find in the market. So we use that, especially for drugs, as drugs we know can attach to the plastics and everything. So we can reduce that and make sure that the effect we see is what the drug would actually do in terms of toxicity or efficacy in the liver. And they are unfortunately not reusable because it's plastic and they come gamma sterilized with the components, and you wouldn't want to have any dried media in between your experiments being like there and compromise and bias your data as well. So it's a single use, but you can use them for over a month if necessary as well. So you can really have extended long-term assays with the same plate as well.
Fantastic, great answer. I think in the interest of time, we'll probably just have one last question here. This person said, it's not clear to what extent one can study the 3D arrangement of cells in this system. Can you just shed a bit of light on this?
Yeah, so I'm happy to cover that. So the essentially in our liver model, what you have is a scaffold-like structure with pores going through it. Audrey showed some images and everything of that. And basically what we are doing really is we're having a mixture of the hepatocytes and Kupffers and stellate cells seeded and mixed within that. But what you still find is, because you've got this single directional flow, you do start to get, the cells being polarized. You start to get the formation of biokinetic and so you do get those structures that you would get in the 3D structure of the cells. And you, as I said, that polarization, because of that single direction of flow, that starts to occur within the system. Now, the reason we do it in those structures, so you end up with these almost like rings of cells through the pores, these kind of 3D structures that occur, is that it's getting the maximum kind of surface area of the cells, having the media go past them. And then that's one of the major ways we keep the cells maintaining the functionalities, is that increase of the oxygen getting to the cells and the nutrients being able to get to the cells. But yeah, that structure, you could probably see it in the slide of the Q&A session at the moment, particularly a little bit on the right. You get these little almost like holes the media can go through past the cells, and that's not a monolayer of cells you see there. That is actually it's got a certain depth to it. So, if you're doing confocal microscopy, you tend to do multiple Z stacks across it to be able to obtain the information you need. So, yeah, hopefully that helps.
Yeah, right. Perfect. Fantastic. Well, I think that's all we have time for today. But Gareth and Audrey, thanks so much for the great answers, both in the presentations as well as the Q&A. It's really been a pleasure having you with us.
Thank you.
Thank you.
Big thanks also, of course, to the audience for joining us today and for the great questions in the Q&A. And so in closing, we hope you enjoyed this Scientist.com and CN Bio webinar, and we'll see you again next time. Have a great day, everyone.